Is a landscape not too far from here. This is sort of like my movement ecology version of the diversity slide that you see a lot of talks start with. But instead of thinking about, questions around how this diversity comes to be, my questions focus on how animals are handling that diversity. How are they adapting to it? How are they generating performance in it? So this is the Champlain Valley. You're looking across at the Adirondacks, and you see all sorts of sources of heterogeneity in the landscape. Spatial, obviously, there's different habitat types, different elevation zones. You can also see dynamism in this environment, which is a particularly interesting thing for folks thinking about movement, right? You see the changing leaves indicating the changing of the seasons. If you look closely too, you also see, maybe not even that closely, you can see human impacts on here. You see agricultural lands, you see developments, and there's even cars down on the road, indicating that the human impacts themselves are dynamic. I find all of this fascinating to think about. What are the mechanisms that are underpinning population trajectories, both natural variability across space and time, but also anthropogenic sources of variability across space and time? So in this talk, I'm going to share with you a few aspects of my research that are aiming at building from these animal responses at really fine scales to these environments, understanding how their behaviors are adapting, how that's changing occurrence distributions, and then also how we can use those types of understandings to reveal the mechanisms that underpin population trajectories, and especially those that are sort of under our control. It could be management targets, policy targets to affect conservation. And then at the end, I'll talk a little bit about things that we can be doing to move this field forward. This is a burgeoning field in animal ecology, the attaching of biologgers to individual animals, and I'll talk a little bit about the potential there and the kinds of things that we can be doing with it. So we'll start here, which is really taking a little bit of a deviation, using seasonal migration as a way to query how animals are interacting with their environments, how that affects their movements, and how their movements in turn affect their experienced environment. And so in movement ecology, you see a lot of things like this, where we're trying to understand how this migration might change a resource curve. And so you can imagine some hypothetical bird going from high latitude, where there's high seasonal variance over time, to a low latitude, and the experienced environment, the sort of individual realized niche, is something like this. It's an integration of all these resource curves across space. We think about this as resource tracking. I'll briefly touch on an example of this from my own research, just so you can get a sense of it, but I'm not actually going to talk too much about this study. I just hate to give a talk and not talk about flammulated owls, which are my favorite species. So this is work that came out of my dissertation in collaboration with the Migratory Connectivity Project at Smithsonian and a tremendous group of collaborators. For this, we put 1 gram GPS tags on these tiny owls, so they're 60 grams. I used to say they're about the weight of a CD case, but that's not really a relevant reference anymore. We distributed these tags across a handful of populations in the West. We tracked them through their journeys, almost a full annual cycle as they return to Colorado. And then we can annotate environmental conditions that we think are relevant to the resources they care about. So in this case, they're an insectivore. And so we annotate it with NDVI and with nighttime temperatures using the MODIS data set. And that's a relative proxy for sort of prey availability, prey accessibility. And we do something like this. So here, without going into a tremendous amount of detail, these gray points on the left are the actual nighttime temperature on the top and NDVI that the birds experienced. And then we simulated non-migratory strategies, and we can look at how they're modifying the realized resource curve as opposed to some alternative strategy. On the right, you're seeing individual comparisons against their simulated counterparts. We say, aha, right, it's resource tracking. And in this paper, we use this to think about how does seasonal migration evolve in the first place, what is needed to support the emergence of such a phenomenon. But this is a really common approach. And it's great, it works. You see the Wyoming mule deer folks talk about surfing the green wave. You see Brianna Abrams talking about this in marine mammals, dampening of resource curves and the flattening of this realized niche. But more recently, what I've been trying to do is increase the sort of complexity with which we view this. The limitations here are that these approaches think about one variable at a time. We do these separate analyses. But we know that as animals are moving around, they're making trade-offs. Some of these resources do not co-occur in space or time, and they're forced to make choices, or potentially to make choices and make compromises in order to generate fitness. And so in this project, we tried to get exactly at that by developing new methods would help us chase this. So this is a big collaborative project. It's a data set from a tremendous number of collaborators organized out of the Max Planck Institute for Animal Behavior. We're tracking 4 species of cranes at continental scales. These are like amazing creatures. Some of them are of very significant conservation concern. We have these 4 species here. So you have common crane up in Europe. You have the demoiselle crane in the red tracks You have black-naped crane doing a really interesting elevational migration, largely in Bhutan, and then you have white-naped crane out in East Asia. They all have unique aspects of their habitat or of their life histories, but they have pretty common habitat needs. Their habitat needs are largely in common. And so what we did is develop a new method that would allow us to explore these potential sources of covariance. And I'll walk you through briefly what that method is and then show a couple results of that. So we grab here what we're calling niche components or niche axes that are relevant to all 4 species. So we have EVI, which gives us something about resource availability. Temperature is obviously very applicable to almost all life forms on Earth, both thermoregulatory expense, but also prey availability. Proportion of crops. So crops for most of these species is a preferred foraging habitat where they're able to access prey most easily. And then proximity to water. Cranes use water as roosting habitat as a predation avoidance thing. So at night, they want to get into these sort of shallow areas or areas shielded by water so that terrestrial predators have less access to them. And what we leveraged is time-ordered principal components analysis, which is a fancy way of saying principal components analysis, but you keep your data in the time order. So what we do is we take all these individual animals, we annotate them to these 4 environmental conditions, and then we can ask about the average among individuals at a weekly time step. There's nothing magic about the week, that's just what we chose. And you kind of keep your data in this temporal order to preserve the time structure in it, and it lets you do a couple of cool things. So the first thing is pretty straightforward. We can look for evidence of trade-offs or covariance patterns among these environmental axes at the scale of the whole year, right? So this is really classic PCA stuff. If we have, loadings that are pulling in opposite directions, it indicates potentially some sort of trade-off. We can also rotate the data back into environmental space with the dimension reduction, right? So you get something that looks like an animal track, but instead of geographic space, you're watching them move around in niche space. You can use this to assess niche conservatism over the course of the annual cycle, and then how these different parts of the annual cycle juxtapose to one another. Then the part that I find most exciting is the PC scores, which is typically something we throw away when we're thinking about principal components analysis. Here, the score tells you for each sample, so every week of the year, how much that sample contributed to the variance captured by that particular axis. So if you line them up across time, the further away they are from 0, the more that axis was mattered to that sample. And you can recover from it temporal patterns of these niche trade-offs. And you can actually think about whether there are individual trade-offs within axes that maybe don't show up in these loading plots. And so we did this in a very simple way where we just say, okay, here's when you're migrating, here's when you're not. And then sort of lay these scores on. And we might see, no rhythm. As you see down here, it might be synchronized with life history events like their migrations, or you might get something like a full annual cycle rhythm. So what did we find? So I'll go through this one sort of quickly. Amongst the 4 species across the annual cycle, there was very little evidence of trade-offs. Really only black-necked crane, the one over here, and the third, you see this tension between proximity to water and EVI. In other words, for that species, it appears that accessing high ecological productivity comes at the expense of safe roosting habitat, at least on the course of the annual cycle. We can also pop these into environmental space, and we're sort of viewing their average migration again in environmental space, and we see actually relatively little niche conservatism, at least with respect to the variables that vary. It's important to note that if you have here, EVI doesn't vary much. So they are probably experiencing a flat EVI across the annual cycle that doesn't necessarily show up down here. But either way, you see these differences amongst these seasonal habitats. You can ask about how spring and fall migration might be distinct from one another or overlapping in niche space. And then the scores, I'll show you examples from a couple of species here, show really complicated synchronization with these life history events. So for many of them, we do see there is this overall seasonal cycle. So despite their migrations, some of them hundreds or thousands of kilometers long, we still see the experience of an annual cycle, which is maybe surprising given the animal ecology literature, or the movement ecology literature, but maybe not surprising at a super high level. And we can also look at what loads onto this, right? So the things that are varying for this species is their proximity to water and their proportion of crops, right? Which is another way of saying that their movements are producing this cycle, right? Because these are habitat choice relevant variables. For almost all the species, The second principal components axis grabbed something like this, so that there were variants that spiked at particular portions of the year. So here, this is the demoiselle crane. We see a tremendous amount of variance associated with the migratory periods themselves. So this axis is capturing variation associated with the migrations. Lo and behold, this one really has to do with temperature. And if I were to zoom back out to that map, which I'm not going to do, what you'll see is that this species crosses the Tibetan Plateau, right? And so there's the second most important axis of variation in this animal's life history has to do with getting over this high elevation barrier over a very narrow period of the annual cycle. Give you one more example from another species. So here's common crane. These ones are largely breeding in the Scandinavian peninsula and then going down to southern Europe in the winter and northwest Africa. Again, we get this seasonal cycle, except in this case here, we're talking about temperature and EVI. And then here we get another life history-based spike in variance. Here's in the post-breeding period, largely associated with the proportion of crops, right? And you can come up with all kinds of hypotheses why that might be. You know, this is the chick rearing time of year. It may be that they're really emphasizing resource-rich or easy accessibility to food resources during this critical time of juvenile development. It could assist with preparing for these migratory journeys. And so we see this really as a way that we can, there's an arrow for it. You know, so this is a way that we can sort of start as a hypothesis to think about how we can understand environmental preferences of these species and how we can understand the trade-offs that those preferences sort of entrain in their life histories. So here, then, I want to touch really briefly on two related concepts about how animals are modifying these environments. Obviously, this suggests that these are really complicated things, not just trying to reduce your annual cycle variance. And so without going into the details, because they're sort of like mathematical behemoths of papers, I want to tell you about a couple of efforts that show that the implications of these complicated changes in habitat and resource availability should have effects on individual performance. So this is a pair of papers that I did with a colleague of mine and of Kate's from University of Colorado, Denver, so Alice and Pierce, where we use dynamic energy budget theory, which is a really cool first principles based theory of how energy moves through organisms. And we extended DEB to account for movement, so explicit costs of movement and benefits of movement. And we can interpret the parameters of these as life history relevant. In a really simple case, the nice thing about DEB is this spot right here, where energy kind of comes in from the environment, stored in reserves, and is mobilized for organismal processes. And the first thing that happens is this split into a somatic stream and a reproduction stream. And it's measured by this parameter kappa. And kappa, that should sort of ring bells. This is classic Stephen Stern stuff, right? This is the fraction of energy invested in self as opposed to reproduction. So there's like literally a trade-off built into the model. There's other parameters in here that you can make this even more complex. But what we were able to show is that one, the seasonality of the environment strongly implies the kappa that's available to you. So not just that it's pulling your life history fast or slow, but it's actually increasing the set of life histories available to you. So in sort of low variance environments, you can have any strategy. In really high resource availability environments, you actually kind of have to be fast pace of life relative to your size. And then Ali and I extended this model with the movement, as I said, to sort of ask about how the resource curves produced by migratory behaviors shake out life history. And the answer, and the reason I'm not going into it in tremendous detail, is because it's very complicated. It's not as straightforward as fast pace of life, slow pace of life. There's been a couple of papers recently sort of positing that migration ought to slow your pace of life down. And that's not what we found. We find that there are these really distinct clusters of energy strategies where, you know, cope is an animal that's really just not moving much. You can buffer, so you can engage in a migration strategy that maximizes your energy intake throughout the year. You can do something in between. You try to compromise between moving, and then it pulls these parameters around, changes your the speed at which you mature, right? So these parameters over here are telling you how fast you mature from development. This is telling you something about sort of how fast your metabolic rate ought to be, things like that. So the point is that it makes this prediction that we should be seeing performance of individual animals tied to this strategy. And I should say that it empirically verifies. When we look at this with empirical data, we see broad concordance with our predictive results, right? That's all simulation. And then more recently, a study I was lucky to co-lead with Nils Linnick at Max Planck again, tried to do this sort of with free living blackbirds. And there's, again, a lot in this paper that I'm not going to talk about, but we have this system where in southern Germany, these blackbirds are co-located during the summer. Some of them migrate south to the Iberian Peninsula and some do not. And what Nils did was develop a system where we can put surgically implanted heart rate and temperature loggers into them. We know who migrates and who doesn't, and then we can measure these like lab quality ECGs of free living birds on their migrations. When they come back, we recapture them and take the sensors back out. There's all kinds of cool bird migration stuff in there, but one of the things we did was a model of the thermoregulatory expense amongst these two animals. And so even though the blackbirds going to the Iberian Peninsula experience this warmer environment, they actually didn't lower their heart rates that much. They didn't really lower their body temperature set points that much. And so there's this difference in energy that needs to be going somewhere to the tune of 18,000 kilojoules on the annual cycle. So this graph here, you have basically watts expended, and you have days of the year across their migratory period with the migrants in red and the residents here. And you can see that there's this savings on thermoregulatory expense that the migrants are experiencing that the residents aren't. Certainly some of this gap between these two curves is taken up by the costs of having to fly, but not 18,000 kilojoules. It's like a 4-day migration. It's a few hundred, it's a relatively short migration. And so our hypothesis then is that this is actually a life history change, that this might be increasing survival and that they're in better body condition, maybe better able to sort of suffer capricious weather events, and or that this is energy that's being saved up towards reproduction as capital breeding. So it's like a quick run through on the idea here is that there's these complicated relationships with the environment. These animals are co-creating that experience of the environment, and that it should matter for fitness. Okay. So, right, what I just said. It's modifying niche and complex and dynamic ways, and that it's altering metabolic organization, suggesting life history. But I started this by saying that we're going to talk about why species decline, and in particular, looking at how humans play a role in that. Human activities are obviously directly affecting animals, but we would also expect them to affect these organism-environment relationships. And that because we know that those organism-environment relationships lead to demographic performance, we would expect that to actually affect demographic performance. And it could be tied to why population abundance goes up or goes down. So I'm going to move to the second part here and think about revealing these mechanisms that are underpinning population trajectories. So what do we know about how human activities affect animal populations? Well, in broad strokes, we've kind of got this down, right? We know it's things like habitat loss and modification, pollution, there's direct exploitation. This is especially important in marine environments. There's disturbance, there are invasive species, and there's climate change. But attributing any of these to any specific population decline turns out to be really, really tricky. So for example, I'm sure most people have heard of the 3 billion birds paper showing that North American overall abundance of North American birds since 1970 has declined by 3 billion birds. Of the fastest declining species, most of them, we don't know why. There's not a clear management action that we can take at a place. We know that it's things like this generally, but we can't actually pinpoint it. Very difficult to study. And so here, I'm going to give you an example of how we can start using biologging to disentangle some of these drivers and actually specify them at the scale of individual animals. So habitat loss and modification versus direct disturbance from humans on the landscape. This is traditionally hard to measure because they're confounded, right? So this is a picture of Shibuya Crossing in Tokyo, a place I got to visit about a year ago, tremendously busy. In general, we see this positive covariance between these two things, right? We are where our stuff is. And so it's really hard to disentangle when we see an impact of something like human footprint index. Is it us or is it the things on the environment? We had an opportunity to study this and break that confound under really tragic circumstances, right? So the lockdowns associated with the COVID-19 pandemic was a dramatic cessation and reduction in human activity that emptied out cities and breaks this confound. And I'm sure that many of you probably saw stories like this in the news about animals sort of re-inhabiting these urban landscapes. Many of them were apocryphal, right? There were no dolphins in Venice. Some of this stuff was totally made up, but some of it wasn't. So there were sea lions in parking lots in Santa Cruz, and sheep in Wales, and wild dogs in Kigali roaming these streets. And so this opportunity caused a group led by Christian Roots to propose a new initiative to use this as an opportunity to understand these dynamics called the COVID-19 biologging initiative. Started in 2020 whilst we were all on lockdown. So I'm a member of the steering committee on this initiative right now. I'm happy to talk more about our initiative model. It's something we're very proud of. It's very open. Anyone can propose projects to use our data set. We have hundreds, I think over 650 data contributors, something like 6,000 or 7,000 animals worth of data, over 1 billion relocations of individual animals in it. And the idea here is that it's a community-led effort that folks can propose sub-projects if you think that there's an opportunity. So if you have an idea, please let us know. I'm going to tell you about one of the sub-projects that I'm leading with my great colleague Ruth Oliver out at UC Santa Barbara. And this is the North American sub-project. Again, a huge data set. It starts with something like 30 million animal observations, I think 3,500 individual animals, right? So it's like one of the largest animal movement data sets ever analyzed. I was joking with folks at breakfast that what we call big data, other people in ecology might not, but this is big data for us, right? And so we have all these animals, and that's the distribution of the observations that we have. One of the reasons we've limited it to North America is because measuring human mobility is very difficult. Most of the papers trying to do this have used proxies for this. So there's stringency indexes, which are looking at public policy and trying to approximate changes in movement. There's some Google mobility reports, but they have really low spatial temporal resolution and sort of a strange interpretation. In the United States, for better or worse, we can actually measure this directly with cell phone data. So this is a product called SafeGraph, which was made freely available for 2019 and 2020 for researchers specifically studying the pandemic. And what it gets us is hourly device counts at the census block group level, which is a weird spatial unit, but it's defined by the US Census. It varies quite a bit. So the census block group is meant to normalize heterogeneity across populations. So everyone has the same number of people in it, but a different spatial area. But we can use this, just like our other environmental layers, to try to understand how many people were literally out and about in the environment at a really fine temporal resolution. And so the workflow that we worked with here goes something like this. We have these trajectories of individual animals of all these different species. So there's our cougar with a radio tag on. And we're going to annotate it to these two measures of landscape modification and human mobility. So I just told you about SafeGraph. Landscape modification, we're using the global human modification layer, very similar to human footprint index. If you're familiar with that, it's an index aggregated from a collection of remote sensing products. So it's basically telling you how urban an environment is, right? So the highest value possible of GHM would be Manhattan, right? And the lowest possible value would be, Bath. And then we annotate it with these other environmental layers. I'm not going to go into model for this kind of thing. It can be kind of complicated. You have to deal with, time series, repeated observations, aspects of phenology. I'm happy to answer questions about it, but we put things like NDVI and temperature in here. And then we characterize the animal's responses in both geographic space. So we characterize weekly space use and then niche breadth. And that doesn't have a unit in it. We're using multivariate normal hypervolumes as a way to estimate niche breadth on a weekly scale. And then we can ask how these two components of human activity contribute to variance in those two response variables. And what we find is maybe not surprising, but should be eye-opening, and that 91% of mammals showed some response to human activities of some sort, and over half of the birds. I'll say right now, most of our birds are waterfowl, and they're weird. They were migrating during the study, which is not great, and they're restricted to wetland habitats. They're typically very well-protected habitats. In the 3 billion birds paper, they're like the one group not declining. So I wouldn't take the 50% as 50% of birds don't care about humans. It's more like waterfowl don't. But we can use this to parse out. So we do this for all these species models, and we can aggregate them. I'm not showing you the species models today, but you can see these aggregated results, which is first, for area size or niche size, how many of the species that we observed responded to either human mobility or landscape modification, or in some way both. And the vast majority responded to both. In other words, if you just measure human development, if you just use something like human footprint index, you miss part of the response, right? That just gets shunted into the residuals of your model. We can also sort of pivot this to ask about how these aspects of human activity, how these components of human activities are driving area size versus niche size versus both. And again, this one here is very, very varied. It's almost equal groupings across these species. This is birds and mammals, suggesting that there are idiosyncratic species-specific pathways to responding to human activities. That there is not a one-size-fits-all to all these species, and that we actually do need to understand at a very specific level what these responses are. The other thing I'll say before I talk about this is we take a subset of our data where we have animals measured in both years and do the same modeling exercise. And we find a strong interaction between the two, which concords with many of our, especially the mammal results. It suggests two things. One, that this is a behavioral response of animals. So these are animals actually responding to what's happening on the ground because it's a year-over-year change within an individual. And the interaction effect suggests that there are outsized impacts of humans out on the environment in the least developed parts of the world, which is probably makes sense from a habituation standpoint, but is an important thing to understand for management prescriptions. This is the same data, again, just showing you the relative effect sizes, lest you think we're picking up on some tiny, insignificant effect that shouldn't affect species populations. This is meant to disabuse you of that notion. It's hard with niche breadth, right? Because that's like pooled variance of a multivariate normal distribution, but there's large percentage changes that we're observing in both directions. And with space use, you know, there's ravens. That's the Yellowstone raven study started by John Marsluff and run by Matthias Loreto. There's a 24 square kilometer increase on a per week basis. These are based on conditional predictions of going from 20%, the 20th percentile level to the 80th percentile level. So the idea here is like, what are the impacts of humans on wildlife generally? And going from a relatively undeveloped, undisturbed environment to that is a huge amount of change. So having these species-specific results that are embedded in here and understanding that the relative components of human activities are important to understand makes it really critical to think about different ways of measuring the impacts of human environments and changing the ways that we're thinking about managing them. So this is a picture of New Haven, where I currently live. I've not yet moved down to DC. This is the kind of environment that we need to be thinking about managing. One of the stunning things that came out of the COVID-19 initiative is that our data is tremendously biased towards wild areas. We have very little tracking data within built environments. There are efforts that have come out of this to try to correct that. So Christian Roots has started this urban exploration initiative directly to try a sort of a re-granting program to try to get tracking projects in these environments. But these are the kinds of places that are going to be where humans are interacting with animals. You know, even if we imagine a world where 30 by 30 is completely successful, it's increasingly hard to imagine. You know, this is what the other 70% looks like. And it would be a shame if we're not taking the opportunity to understand and manage in that other 70%, more than a shame. Especially given the current rates of extinction, right? We all know there's something like 1,000 to 10,000 times above the background rate. And again, highlighting this 3 billion birds paper, common species that we don't think of as on the brink are declining rapidly. I was recently putting together a proposal looking at eastern meadowlarks, which is not a species we think of as sensitive. Their population has declined 80%. Chimney swifts, it's a synanthropic species. Their population half-life is something like 14 years. So this really is a critical problem, and it's a critical problem overlapping built human environments. And I also I think it's important to pause and recognize that these are human problems too. It's easy for someone like me to think like, we got to save these species, like I'm motivated by it, but healthy biospheres are also fundamental components underpinning healthy human societies. And that's why things like life below water and life on land are UN sustainable development goals, right? Not sustainable make nice parks and bird watching goals, but how to develop human landscapes. So that sort of gives us a mandate to think about how these human activities are contributing to biodiversity loss. Hopefully, I've already convinced you with those data that we need to disaggregate that a little bit and actually start to think about how the components of human activities are contributing to biodiversity loss. And hopefully, I've at least started to convince you that we need to do this on a species by species basis, that thinking about declines as these big biodiversity indices don't actually capture enough of the heterogeneity that we see among species. And so we need to think about species declines. So how do we measure that? I'm going to give you population biology 101 here, just in case anyone wants a refresher. The idea here is that when we see a change in abundance over time, it's actually the result of 4 confounded processes, right? We're really good at measuring abundance for many taxa, not all of them, but we have good monitoring. There's citizen science, like all the stuff in GBIF. In birds, we have breeding bird surveying versus bird count. We can track these abundances. But understanding the actual biological processes that lead to these changes is actually quite tricky, right? So changes in abundance arise because you have changes in birth rates, immigration rates, death rates, and emigration rates. Those are the ways animals get in and out of a population. When you have unmarked animals, you cannot observe those 4 vital rates. Right? You don't know which one because they are completely confounded. You could have tweaked any single one of those in any sort of way to have made this abundance decline. So these occurrence-based data sets that are getting us things like species ranges and range shifts and abundance declines are insufficient to actually diagnose the things that are causing species to go extinct. And I'm going to propose today that animal tracking and biologging generally can help us close these gaps. Part of the reason is that biologging marks animals. So this is day 2 or week 2 of population biology 101. The way that we disaggregate, or at least partially disaggregate those vital rates, is by marking animals, trying to observe them over time. And with that, we can start to partially observe some of these demographic processes. So you can understand if an animal went missing because you didn't observe them. It might have been that they were unavailable, but there are robust methods for this. I don't want to criticize these because I still use them myself. There's a whole line of my research that I'm not showing you today that uses this kind of data, but there are limitations. The observations are restricted to the study area. You have very low resolution in your observations of the individual animal. It's very hard to get sort of auxiliary information about that animal. And so you end up with coarse temporal resolution. You end up with not really being able to tell if the animal died or just left. So there are real challenges here. Animal tracking, on the other hand, solves many of these problems, or at least has the potential to. One, you're not really restricted to a study area. So the problem of emigration largely can go away. We can actually start to disaggregate those 2 vital rates and break that confound. We can tell when an animal has left versus when it has died. Things like births and fecundity are emerging. We certainly can't do that as well, but there are emerging methods to do that, both through sort of behavioral classification systems and also direct biologging tags that can sense that. In our highest quality data, we can actually just detect the mortality directly. We have in large animals mortality switches that can tell us when an animal died. And so you're not just getting these vital rates, but you're also getting fine-scale behavioral information leading up to that. We know the environments the animal is in, we know the kind of movements it was making. You can put heart rate monitors on it and understand what its heart rate was doing. Use accelerometers to get foraging attempts. You put cameras on them to get foraging success, right? There's all this rich behavioral information that's really the purview of movement ecology proper that we can bring to bear on these population biology questions that are so urgent for conservation and management. And see if I can get this to play. Yes. And there's a ton of it. There's a ton of this information. So this is an animation from Sarah Davidson's paper, the Arctic Animal Movement Archive. This came from the NASA Above project. This is a multi-species, multi-year tracking effort with thousands of animals. Most of this data is open source and available. And you can see that this data is obviously giving us something about how these animals are moving, right? I mean, I'm just sort of mesmerized by watching them go up and down with the pulse of winter and freezing sea ice. But we could also use this to get demography and associate it with all that stuff. Each one of these is by definition a repeated observation of an individual animal. And so therefore it is suitable for demographic analyses. There are tens of thousands of animal tags on right now that are feeding in live, not even need to be captured later, not even need to wait for a data upload, but are directly transmitting to satellites. Something like 65,000 tags right now doing that. So it's a tremendous opportunity to use these data. And so putting those 2 ideas together, we can obviously use this to really pinpoint where these mortalities are occurring, when they're occurring, because we don't expect them to be distributed across the annual cycle. If somebody studies bird migration, this is something that's hammered into us. We think that their different periods of the year have differential risk profiles. So it might be really important to understand when these mortalities are occurring. We can get better at assessing approximate causes, and then ultimately how to get at the anthropogenic drivers that are pushing those causes and leading to those vital rate changes. And then the last advantage here is this how, and that we can get these temporal time series. So most of our methods right now synchronize those mortality assessments with changes in the environment at the time of mortality, which is not a realistic assumption in many cases. Many of these effects may be chronic, where there's some driver that's elevated throughout the lifetime of an individual animal that's leading to its death, or episodic, or lagged, right? This is in the bird world, we call this a carryover effect. And I want to give you an example of some research where we were able to do this with animal tracking. So this is a study that I was happy to be a part of, led by Nate Cooper at the Migratory Bird Center, one of my colleagues, as well as Clark Rushing and Scott Sillett and Pete Mara, where we used two different methods for two different long-distance migratory species to try to understand the drivers of mortality in them. I'm not going to talk about the black-throated blues today, because we actually did that with a sort of a clever approach where you use old school bands at two different linked populations, and you can suss out the full annual cycle survival rates. It's a method that Scott Sillett pioneered in the early 2000s and Clark has extended. But with these Kirtland's warblers, this is one of the rarest warblers or one of the rarest birds in North America, recently delisted, but still extremely small population size, entirely reliant on humans for management for the species to occur. And they're tiny, they're 16 grams or something like that. So they can't carry most of our tracking devices. They winter down here in the Bahamas. Nate set up a study system on Cat Island, this tiny cactus scrub island in the Bahamas. And then he has a breeding season study up here in the jack pine in northern Michigan. We used an emerging technique of automated radio telemetry. It's the MODIS system. So it's sort of ground up, organically built, like anyone can put out a tower for your own project, but it also catches everybody else's data, and the tags are tiny. They're even tracking monarch butterflies and dragonflies with, right? And it's just an old school, it's a coded radio tag. It goes by a tower, anybody's tower will pick it up, and you know which individual went by, you get a timestamp, you get an XY, and you can turn this into movement data. It also looks a lot like mark recapture data, except that it violates all the assumptions. Kirtland's warblers are great because they all go to that one place in Michigan. So without getting, again, happy to talk about the details of how you fix a Cormac-Jolly zebra model to do this, but the biggest challenge is that this idea that if they left the population, we can't attribute that to a mortality. And when you're talking about moving animals in an irregular network, that's like a huge problem. But we manually survey 97.5% of the known breeding grounds of the species every single year. So permanent emigration is almost zero in this species. And it allows us to deploy this method on this species. And to do so, what we did is we get like home range size down in the Bahamas. We can associate environmental conditions. We use EVI, which is a proxy for the sort of environmental productivity and the moisture, which is known to be an important factor for birds overwintering in the Caribbean. And then we can estimate survival as they move north. So we estimate survival during the winter before they've left the Bahamas, on their trip up to Michigan, and then up on the breeding grounds. And here's what we find. So here you have weekly survival rates on the y-axis, and you have EVI on the x-axis. So higher numbers are wetter, and then the different colors are showing you the different seasons. And there's two things here. The most obvious is that spring migration is riskier. We saw significantly lower survival rates during the spring migration, which we've suspected. For those of you not initiated on how you track small animals, like this is the first direct estimate of survival of a small migrant bird, right? So most birds, 60% of birds, we can't track with the cool GPS tags. So this alone to confirm this is an advance for us, but also that this carryover effect, we see the significant effect of winter EVI that propagates not just survival in the winter, not just while they're migrating, but follows all the way up into the breeding season. So your wintering conditions change your survival outcome on the breeding grounds. And then we can pop this into a population model to make predictions about what we might see at the population levels. We take published estimates of the other vital rates from the literature, from sort of tried and true wildlife biology studies, and then we can take our model estimate and plug it into, just a simple Leslie matrix, and then vary EVI to see what would happen as the Caribbean dries, which is what we expect, right? The Caribbean should be moving, in this case, to the left of this graph. And here the dashed line is lambda, your population growth rate. If you're below 1, your population is declining. And as you move towards drier environments, the population growth absolutely collapses. This is critical information. The success of this species being recovered has to do with the production of fire-dependent jack pine in Michigan and the removal of cowbirds. No amount of that management intervention fixes this problem. And so if we don't see that, we're going to keep using the tool that we have that we think has worked and would have no chance of success for this species, right? In this way, what we're able to do is directly observe what has the potential to change population abundance, because we've observed the mechanisms, we've linked it to their behavior, we've linked it to their environment, and we can make real recommendations for the species that are targeted, have a high degree of success, and are efficient. But we want to build it up more. Like I said, there's a lot of data out there, and people are collecting more and more every day. And so Ruth Oliver and I have launched a new project under the COVID-19 biologging initiative that's trying to take a mortality approach to disentangling. So sort of combining this last idea with the COVID project that I showed you, the North America project, to try to understand how different components of human activities are driving patterns of animal mortality. And sort of a long-term vision would be, we produce these atlases of animal distributions, there's atlases of migration and migratory connectivity. You could imagine atlases of vital rates, right? An atlas of wildlife mortality, the death atlas, right? Where we understand the spatial distribution of these vital rate risks and the attribution to environmental and anthropogenic causes. So we're building a global data set. This is my little advertisement. If you have animal tracking data or know someone that does, we're always trying to build this up. We're collaborating with Fran Cagnaci, who runs the Euromammals project. They actually integrate field validated mortalities with their tracking data, which is really, really cool and important. Okay. So hopefully what I've convinced you of is that human activities are in fact changing these organism-environment interactions, and that these animal responses to these things may be connected to the global loss of biodiversity writ large. And then hopefully convinced you that individual-based ecology, in particular animal tracking and biologging, can help shed new light on these mechanisms. That gives us both basic ecological insights, but real tremendous potential for applied insights. And so the last couple minutes, I just want to talk about the opportunity that we're facing as this field grows. To unlock the potential, we're going to need to continue to take advantage of the things that have been rapidly developing in this field over the last couple of decades. And that's new technology, new tools, new communities, and that gets us to new and old questions. Roxanne Beltran has a tremendous paper that came out a couple months ago in Tree, highlighting the opportunities for this kind of thing to also unlock new insights in fundamental biology and ecology, right? We're measuring things that are previously only done in a lab on free living animals. We're measuring assumptions about vital rates and density dependence that were really the purview of theoretical ecology. Now we can measure it. I want to highlight the advances in technology for animals, just to give you a picture of the trajectory of this field. So this is probably the first extrinsic tag ever put on an animal. It's called a field stork. This is a white stork, sort of the national bird of Germany. It was found in Germany alive, collected for a museum in the 1820s, I think 1821. This bird had apparently flown to Africa, unbeknownst, right? The Western Europeans did not know where white storks went for the winter. Barely survived an attempt at hunting on its wintering grounds, and somehow made it back to Germany with that arrow in its neck, was collected. They take the arrow to anthropologists who know what part of Africa that must have come from, and all of a sudden you have 2 points on a map. This happened 27 times, enough that they named them, right? So they're called the field storks, and you can find them. There's a couple museums that still have these. You fast forward to the last few years, and the technology that we're using, in addition to some of the things that I've shown you, is stunning. So this is a study that radio tracked a moth on its migration and found it course correcting for winds, right? So they're repeating the Bill Cochran and Martin Witkowski study with the van in Indiana tracking thrushes, but they're doing it with a moth, right? This is Nils Blackbird logger again for the energetic modeling that I showed you, right? This is a tiny ECG and temperature readout that we can put in a 100 gram, it's the size of a robin, right? This is an Icarus tag, also pioneered by the Max Planck animal behavior folks. This is the 5 gram one. They're working now on a 3 gram and a 1 gram one. This gets you GPS location, accelerometry, temperature, and it uploads to satellites. And so it's tremendously powerful for we don't have to catch this bird a second time. to follow them. And then a line of research that I didn't talk about today, looking at the drivers of morphological change in birds, using these MODIS technologies, right? This is an old technique. Radiotelemetry is like pretty tried and true, but we're finding new ways to put this on birds, and using this network, we can do manipulative experiments and track juvenile birds we have no hope of ever capturing again. We can still follow their movements on continental scales. And all this means that we have a tremendous amount of data. This is already out of date. This is a few months old. There's 6 billion records in MoveBank alone. That's just one repository. It's our biggest, but there are others. There's almost no marine data. They use different repositories. There's billions of records of these animals that we can leverage right now, even if we were to never put another tag on. And the last thing I'll leave you with is sort of zooming this up to scale. So I was at a meeting down in Washington, D.C. last week where the Smithsonian, in partnership with a handful of institutions here, in particular Wild Labs and NASA Jet Propulsion, are trying to figure out a way to zoom the other way. So I talked a lot about the mechanisms within individual animals, but you can also aggregate this stuff to get real insights into biodiversity processes. And so we're starting a bond, biodiversity observation network. These are critical networks. You've probably heard of GeoBond. There are other thematic bonds. They serve up sort of aggregated metrics and indicators to support global level policy initiatives like the Convention on Migratory Species, Convention on Biological Diversity. And we think that there's tremendous potential to develop movement-based indicators and metrics that help us both understand the things that we're already monitoring at a more mechanistic level, but also to get us at new things. Like, it would be great if we had indicators that were actually measuring the reasons that species declined rather than coarse proxies for those. And so this is something that we're very excited. It is just launching now, and we're sort of looking for new partners and folks to aggregate up, but there's a lot of energy behind this. And with that, I'm happy to stop and take questions, and thanks again for the invite. It's tremendous to be here. Yep.
Thank you. I could ask you 1,000 questions. I'm going to try to maybe ask 2 on the slide of that blackbird.
Yeah.
One was the difference in the energy expenditure, whether it was migrating or not.
Yep.
And so essentially those are like alternative stable history strategies. What's the if there's that huge energy savings of migrating, that's right?
Yeah.
What's the kind of trade-off there that?
Right.
Like,
Yeah.
all those kinds work. So why is that?
Yeah. And so I'll make it. Oh, sorry. The question was with the blackbirds, right? Like, those look like they are potentially 2 stable strategies, and why do we have some mix of both, right? So it's a great question, and it's sort of that's the fundamental question in the paper that I find interesting, that I think we make. So the historical explanation for why you have these we call them partial migrants, is that it's basically a density dependence thing. And so you get this trade-off as the effects of density dependence get to a certain point, all of a sudden it tips over, like the nth individual, it is advantageous for them to exhibit a different strategy. But that's not it with the blackbirds necessarily, for a couple of reasons. One, they're facultative partial migrants. So individuals may migrate in one year and not migrate in another. And I think that difference in the graph is actually the explanation. My suspicion is that actually it is a modulation of the life history strategies. What they're cueing into to choose that strategy is unclear, because it has to be behavioral. The other thing I'll note is, like, in that study, we talked about, we look at their ECGs, and there's a couple things that are really interesting. So the dynamics of how fast their hearts are beating across the 2 strategies vary. But if you add up the heartbeats over the year, it's the same. So there's that's why that graph works. When I model the thermoregulatory energy, the difference there has to be a deficit, because they had to have spent the same amount of energy, assuming the hearts are the same size, which is, I think, a good enough assumption. We also see them prepare for the migration with their heart rates before fall, but not spring. So you can hold these birds in the hand the day before they migrate, and you can't tell, right? This is not a huge migration, so they don't engage in hyperphagy and like blow up 20% and huge fat scores in the furcular hollow. The 27 days before migration, the blackbirds that are going to migrate depress their heart rate at night, and they lower their body temperature set point. And we assume it has something to do with, allowing them to anabolize tissue or fat to pay the costs of the migration, which is part of the reason we don't think that energy deficit is actually paying for migration. So they're cueing in something months before they do it, or at least a month before they do it. Yeah, we don't know. That's something I'm trying to drive at. So a colleague of mine at Smithsonian and I are trying to figure out if there's a US system that we could use equivalent to that and do manipulations with. So like this summer, we're going to put out tags on eastern bluebirds, which we know are partial migrants. We don't know if they're facultative. And if they are, then we can do manipulations. And then the group at Max Planck, so that's out of Jesko Parteki's group. He's one of the group leaders under Martin. They've been doing some translocations to try to suss some of this out. Sorry, I gave you the long answer. Yeah. Yeah.
There's kind of just like staff in urban, like the appearance of animals in urban environments, even like during the COVID period where animals were kind of just everywhere.
Yeah.
There was no barriers to them. It's nice. Is there like any further research going on or anyone doing work to try to address that information?
Yeah. So this was a big thing that came up last week at the MoveOn, right? I mean, if you're trying to do a biodiversity observation network to assess global policy, you really ought to have coverage over the environments that affect humans. Again, I think like it's important that we take a humanist approach to this. It's not compelling, sufficiently compelling to the general public that we just protect species for their own right. And I don't think we need to sell anybody on that message. We know it's important. So there's a couple efforts to do that. So Christian Roots has started a pilot of this urban exploration project. It's in partnership with National Geographic. And the idea here is a really ambitious, essentially a re-granting scheme, where we try to address these gaps directly by feeding money into project level teams that want to do something in some portion of the built environment, kind of along a urban rural gradient. And then they're sort of committed to data sharing for these aggregated synthetic products after the fact, and you can co-design all these things at the same time, right? So there's like a year-long co-design process kicking off. But the ambition for there is huge. I mean, they're envisioning, putting up 10,000 tags across 250 projects. So that's, I think, a really cool way of targeting that particular gap. The other thing we need to do is just actually get a better handle on how bad that bias is. So one of the things that was remarkable is like finding the data is really hard, right? If it's on MoveBank, then great, I can query MoveBank. 8% of MoveBank is public. An unknown percent of data is on MoveBank or Animal Telemetry Network or one of these others. But there's a tremendous amount of data that exists nowhere discoverable. And we were able to discover some of it by just, emailing our friends and putting up, the paper in Nature E&E that Christian wrote. Like people contact us for that. But that's not sufficient. I mean, agencies are collecting data. So much of this stuff is in hard drives and people's drawers. And I have a history in environmental consulting. So I've used some of this data where they're like, Bob collected that 20 years ago. And let me see if I can find the right closet. That's unacceptable. These data are, there's an ethical obligation to the animals. These data are expensive to collect. It takes a tremendous amount of training and permitting. The tags are expensive. So one of the things that we've proposed doing is a registry. So, one of the hesitations, people don't love sharing this data. It's getting better, but there's a hesitation to even put it up anywhere because they think they're going to get scooped. So our idea is to have a tag registry that just says you deployed a tag. Your data it's just the metadata, right? We do this with cars, right? We know every car in the country. I don't know where you drove, but I can figure out who has a car. And that's a start, right? Even that metadata would really start to reveal the biases. So we're working on that. It's something that Christian has proposed, and I think there's a good home for that at Smithsonian. The thing we're struggling with is how do you get people to use it, right? What's the carrot? What's the stick? You can imagine using permitting, funding. You go through the manufacturers. Modis is a cool example. So in order to buy a Modis tag, you pay the manufacturer your Modis registration fee, and you agree by default to the data sharing agreement. So it's just a privilege of using everybody else's towers is that you're in on the network and it's all unified and there's a central repository. So yeah, we're working on it, but it's a big, the biases in biodiversity data in general, and we're not immune. Yeah.
Real quick follow up to that. So the BBL should have data on everyone's birds, right? That should be part of your application process, permitting process.
Yeah. So the question is, does BBL have this? Yes, to an extent, although I'm not sure that the publicly available BBL has auxiliary tagging data on it. Yeah. But I was talking to some folks last week. I think that's the place we start. It's like, just get BBL and BBO to do this. If the United States and Canada, all the birds tracked, we end up going on a repository. That's a start. And then we build momentum, and that's a good central place. And the MoveBank folks are totally on board. They're like, we'll build you the data. This is not like, the data is not hard. It's just it's a table. We just need to get people to fill it out.
So actually, I had a question, sorry, about the warbler.
Yeah.
stuff. So, sorry if I missed this, but why was fall migration not included?
Oh, the tags died basically. So we those tags we deployed, we censor the study, so there's a couple birds we get fall migration and post breeding. We would love to get it. So with the black-throated blues, we can get fall migration using Clark and Scott's method. But so we have a couple grants right now to actually look at that. So we're trying to figure out, some of these other pieces of the annual cycle that we usually kind of treat as blurry or lump in with other things when we do full annual cycle population models. We're actually trying to figure out what is the pairing rate. Like we just ignore that with birds a lot of time. Juvenile survival is this completely hidden thing because it's so hard to track birds that are juveniles. Their mortality rate is high. They're not coming back to your study sites. So you have to put something on them you don't have to capture again. And so we're working on it is the short answer. Yeah.
In a lot of mammal species, you get a diurnal nocturnal displacement relative to human behavior.
Yeah.
And I was wondering whether that, in the studies where you had the anthropogenic data, you've been able to compare how that changed compared to the more common adaptation of species to nocturnal pollen.
Yeah, so the question is like, do we see a shift in diel cycling, a shift towards nocturnality? With our data, we could totally look at that. We haven't because there's other groups doing it. So Caitlin Gaynor up at British Columbia did a really cool study on that. And there was a more recent review in Tree by, I forget his last name. His first name is Neil. It'll come to me looking at that. Yeah, with mammals, that is a super common outcome.
But if you compare it to the anthropopause, because obviously you get these species quote showing up where they didn't show up before, but was that they showed up there?
Oh, right. Yes. So, right.
Daytime, you never saw them.
So with what we were looking at, we're using biologging, so we don't have that bias, but we do know if there are shifts in night and day. We don't look at it in that paper, but there are other papers doing it. And like I think Chris Wilmers had the study with the cougars in California, and I think he saw that they went more diurnal. Because and cougars are a great example of that because we know that they engage with human habitats all the time. They just do it at night when we don't see them. And so they definitely shifted towards diurnal. And I believe there's another sub project that is looking at that explicitly. We have sub projects on that. Road crossing is another sort of low hanging fruit. So there's a group based out of Fran Cagnacci's group looking at, did we see a change in road crossing behavior or not, and which animals exhibited it? Yeah.
Really interesting talk. Great to go out on everything you're doing. I also had a question about the blackbird studies. It's kind of two-part. The first one's pretty short, but I was initially curious what percent of these birds are actually migrating, and then secondarily mentioned it's a facultative thing where some years they go, some years they don't. If it's a small percent, presumably there are possibly you could just not observe migration in birds that migrate. Do we know how often birds like turn off migration because they're facultative and conditions are good for a couple decades? Yeah,
so good questions. Across space, there's a gradient. So, and this is true for non-facultative migrants too in general. So, if you go up into Scandinavia, they all migrate. You go down to Spain, nobody migrates, and there's just a clean gradient across. Where the study population we worked with in southern Germany was close to 50-50. I think there were a few more residents than migrants. In terms of interannual variation, that's a great question. I don't think anyone's looked at that. That's so like our idea with the blackbirds is like, or the bluebirds is if they were facultative, could be then manipulated, right? So if I supplement food, can I change that, that proportion? And then same thing. I mean, I think that's where, I don't know all their results super well, but I think that's where Jesko's group is using these translocations to try to get at this question of like, if I take a bird from a group that shouldn't be migrating and move it, what will it do? It's an end around a similar question.
We have hit noon, so we'll pause for now, but if you have more questions for Scott, we will be at lunch and happy hour. So we'll go another round.